Hi guys, I'm Per, and in this video I'm going to introduce to you a type of cell division, and that is mitosis. The functions of mitosis include allowing us to grow and as well as healing our injured tissues. Before we get into the stages and the processes of mitosis, I think it's important that we review the cell cycle, because there are things that need to take place before a cell can divide. And I also think that this will help us see and understand a more detailed and a bigger picture of how a cell divides in mitosis. So to start off, I've made a diagram of the cell cycle. And the cycle is going clockwise, this way. The first phase of the cycle is known as the G1 phase. At this phase, all of the contents inside the cell start to duplicate themselves. So things such as ribosomes, mitochondria, Golgi apparatus that are in the cell, they will make a copy of themselves. The only things that are not being duplicated are the chromosomes, which are of course inside the nucleus of the cell. So cell contents duplicated except the chromosome. So in other words, during this phase, the cell is growing. And we say the cell is growing because the vast majority of the stuff that is contained inside the cell will be doubled. The next phase is the S phase. The S actually stands for synthesis. So once we enter this phase, each of our chromosomes start to duplicate themselves, and the way they do that is through a process called DNA replication. Here I'm not going to go into how the DNA is replicated, but essentially each of our chromosomes, for example, we have a chromosome here, and this chromosome will go through the process DNA replication and will turn into a structure that looks like this. So yeah, so this new structure that we have, this new chromosome that we have, is different, and we can see that it has two chromatids. So one chromatid here and the other chromatid here. And these two chromatids that we have, they are identical, and we call them sister chromatids. Sister chromatids. And again, they are called sister chromatids because they are 100% identical. So during this S phase, we have all of our chromosome duplicated. So chromosome duplication, and this is done through the process DNA replication. Following the duplication of the chromosomes in the S phase, now we can move into the next phase called G2 phase. There are important activities and various processes that take place prior to this G2 phase. But the most important process is the DNA replication. chromosome contain all of our genes, and it's critical that the genes are encoded correctly. If a gene is not encoded properly, a lot of things can happen, and sometimes those things can be bad things. For instance, the gene may become dysfunctional, and that may lead to the organism being susceptible to a particular disease. So the main purpose of this phase is checking for errors and mistakes. Checking for errors and mistakes in the process DNA replication. This is also known as DNA proofreading and repairs. And if you're curious about the proofreading mechanism, there's basically a particular group of enzymes called DNA polymerase that recognize these mistakes during DNA replication. For more details on the proofreading, DNA proofreading, that will have to be as a separate video. So, in addition to this phase, toward the end of this phase, the cell prepares and gets ready to divide. So, cell prepares and gets ready to divide. So now, and by the way, the phases G1, G2, and the S phase, they are the phases that we call interphase. Interphase, which is over here. We call them interphase because they are the stages where the cell is not dividing. After this G2 phase, we move into the last and final stage, and that is mitosis, which is what we're here for. But one last thing, cells need to divide and proliferate in order to heal themselves, but they don't always and constantly divide unless there's a mutation, such as in cancer cells where the cells constantly divide without control. And in theory, and basically, they can proliferate or divide endlessly. So let's assume that we have normal cells, which means there's no mutations. The cells that are not dividing will stay in the stage called G0 stage, which is right over here. Going into mitosis, there are 5 phases. The first phase is prophase. Here we see a cell, and inside this cell we have an enclosed organelle. Organelles are like the structures inside a cell where each one has a specific function. And this particular organelle is called the nucleus. As you can see, it's the biggest organelle inside the cell. And inside this organelle, we see this thread-like structure that's made out of nucleic acids and proteins. We call these structures chromosome, which we're already very familiar with. As we can see, during this phase, there are 4 chromosomes. And the chromosomes are lined up alongside one another in pairs. Here, we're only going to use 4 chromosomes for simplicity purposes. For example, there are a total of 46 chromosomes in every single cell of the human body. The only exception is of course, in the reproductive cells called gametes, which have the number of the chromosomes. Another example is fruit flies. They have fewer number of chromosomes than humans, with 8. What the fruit flies have in common with humans is that they are both diploid. What that means is that the chromosomes come in pairs, and so during this phase, prophase, each one of the chromosomes is going to find its pair inside the nucleus. So one pair here and the other pair here. Once they find their pair, they will be physically beside one another. So we call them homologous pairs. So one homologous pair here, the other homologous pair is here. Just pair or chromosomes. Homologous pairs or homologous chromosomes. They are homologous because one is from mom. So let's say this one is from mom, and this one is from dad. And this chromosome 2 here and this chromosome 3, they will be non-homologous chromosome. Once all the chromosomes line up alongside its homologous pair, the next phase is metaphase. Where we see the nucleus envelope has broken down and all the chromosomes move toward the middle of the cell. So here, the chromosomes line up in a line from one end to the opposite end of the cell. What we also notice is the appearance of these lines that we call spindle fibers, or spindle microtubules. Their job is to attach themselves to the structure near the centromere of the chromosomes. That structure is actually a type of protein. So if we zoom in to the chromosomes, and by the way, this will require a special type of microscope because it's really, really small. This is what we see, the protein called kinetochore in purple here. In purple there. Is located just on the centromere of the chromosome. The centromere is the constricted region of the chromosome, of the this condensed chromosome, located in the middle right here. So the function of this purple kinetochore proteins is to be the attachment site for the spindle fibers. So during this metaphase, the spindle fiber will attach themselves here, like so, to the protein. And this is essential for accurate chromosome segregation. So in metaphase, the nucleus disappears. The chromosomes line up along the equator of the cell. Then the spindle fibers start to appear and attach themselves to the protein called kinetochores. The next phase is anaphase. This is when we start seeing the sister chromatids being pulled toward the opposite end of the cell by the spindle fiber. So this is one chromatid and its sister chromatid is here. They are being pulled toward the opposite end of the cell, like so. Following anaphase, the sister chromatids keep moving toward the end of the cell, while the cell pitches in the middle, and it looks something like this. We call this phase telophase. The last phase of mitosis gives us two completely separated cells, and it's cytokinesis. Note that these two cells are 100% identical. They are also known as daughter cells. Two identical daughter cells. So one daughter cell here, the other daughter cells here, and they are completely identical. Also understand that each of these two daughter cells contain the full set of DNA. In this case, it's a human cell. So one cell here is a diploid cell. So diploid. And this one is also diploid. You can also tell by looking at the number of chromosomes inside the nucleus, here and here. The process started with 4 chromosomes at the beginning and ends up with 4 chromosomes again in each of the new daughter cells.